All right, welcome everyone today. We're very excited to have four experts in the field coming to us from Stanford University to talk about health equity disparities and challenges in type 1 diabetes. And the Stanford team consists of Dr. Danny Chou, who will discuss novel insulin formulations in context of this talk, Dr. Rayhan Lal, who just discussed new diabetes technologies in context of the discussion, Dr. Ananta Addala, who will talk specifically on health disparities in diabetes, and finally, Dr. Gloyn, who will discuss genetics and islet function. And so I'm just doing a quick bio for each of you. You're all very well-versed and expert in this field, and we really appreciate you joining us today. I'll start with Dr. Gloyn. Dr. Anna Gloyn joined the faculty at Stanford University in February 2020 after 16 years at the University of Oxford. And she, while at the University of Oxford, she completed her DPhil at the University of Oxford under the supervision of the late Professor Robert Turner. Her postdoctoral training was carried out at University of Exeter under the mentorship of Professors Andrew Hattersley and Stan Allard at the University of Pennsylvania in Philadelphia under the mentorship of Professor Fran Meshinsky. At 2004, she returned to Oxford with Diabetes UK RD Lawrence Career Development Fellowship and established an independent research group focused on understanding beta cell function through the investigation of genetic variants causally implicated in monogenic diabetes. And in 2011, she was awarded the prestigious Wellcome Senior Fellowship in Basic Biomedical Science, which she successfully renewed in 2016 and transferred to Stanford when she relocated. So she's got a deep understanding of the scientific background of type 1 diabetes. Dr. Danny Chou is Assistant Professor of Pediatrics, Endocrinology, and Diabetes at Stanford University. He received his PhD from Harvard, working in the lab of Professor Stuart Schreiber. His PhD research involved the identification of suppressors of cytokine-induced apoptosis in pancreatic beta cells. He then moved to MIT, where he was the JDRF postdoctoral fellow in the Department of Chemical Engineering. He worked under the guidance, letting more people in. He worked under the guidance of Professors Bob Langer and Dan Anderson, focusing on the development of glucose-responsive insulin derivatives. And he started his independent career in the Department of Biochemistry at University of Utah in 2014. While there, his research focused on protein and peptide therapeutics for treatment in T1D and other human diseases. And then in 2020, he moved his research to Stanford to continue efforts in developing novel insulin therapeutics. And his laboratories received funding from NIH, DoD, JDRF, and ADA. He's also received recognitions, including an American Peptide Society Early Career Lectureship, Boulder Peptide Society Young Investigator Award, JDRF Career Development Award, Vertex Scholar, JDRF Postdoctoral Fellow, and ADA Junior Faculty Award. Dr. Ananta, so shifting gears to Dr. Ananta Addala, she's a pediatric endocrinologist and physician scientist, addressing disparities in pediatric type 1 diabetes management and outcomes. And as a physician with a background in pediatric endocrinology, epidemiology, and behavioral health, she aims to build an evidence-based approach to addressing T1D disparities by systematically evaluating youth, family, provider, and system-level barriers to optimize diabetes care in youth from low-income, socioeconomic, and racial ethnic minority groups. To date, her publications have demonstrated that the disparities in pediatric T1D by socioeconomic status are worsening in the U.S. Provider bias against public insurance is common, and public insurance-mediated interruptions to diabetes technology adversely impact glycemic outcomes. She's also been leading the efforts to improve justice, equity, diversity, and inclusion in research at Stanford University through her leadership at Stanford Pediatrics Advancing Anti-Racism Coalition and the co-chair of TrialNet's Underrepresented Minorities Outreach community. So far we have three incredibly important, laser focused scientists and clinicians addressing T1D. And then finally, Dr. Lal, he grew up in the East Bay and he's had type 1 diabetes for 30 plus years. He studied electrical engineering and computer science at UC Berkeley, go Bears, with the hope of applying my knowledge to diabetes technology. The significance of clinical practice became clear to me after my siblings also developed diabetes, and he has devoted his life to advancing the care of diabetes in people of all ages. So I'd like to welcome all four of you. You bring a really unique perspective and well-rounded perspective to what's going on with diabetes right now in the US and by extrapolation to the world, as it pertains to, clinical sort of experience and patient experience and diversity and outcomes. So if you want to, I think Dr. Addala is going to start. So if you want to start and take it away.
Right. Welcome, everyone. I'll actually hand it off to Anna first to start as to kick us off, and then I'll take over. But yeah.
Great.
Okay. Well, good morning, everyone. Thank you, Monica, for the introductions and also the opportunity to speak with colleagues this morning around something that we're all very passionate about. And it seems very timely to be doing this in Diabetes Awareness Month. So when Monica reached out and asked if we'd be interested, I immediately thought that this would be an opportunity for us to share with you some of the exciting work that we're doing at Stanford. And I called upon my three wonderful colleagues to share the platform so that we could tell you about different elements of work that are happening at Stanford. Ananta, if you wouldn't mind just forwarding the slide one, that would be amazing. Some disclosures, leave it up long enough for people just to drink that in and flick over. Okay, so what we thought we would do this morning is really try and give you an outline of the exciting times that we're in for diabetes right now, particularly type 1 diabetes. When I was a PhD student, there were really only the options of treating your diabetes with insulin, or if you had type 2 diabetes, sulfonylureas, diet, metformin, or insulin. And really, we now have so many more options, and that's exciting, but it also means that there's a greater opportunity to think about tailoring the right treatment to patients, and also thinking about access to those treatments. And what we'd like to do today is really tell you about some of the ways that the work that we're doing is trying to address those questions. My background is in human genetics, so I'm hoping to give you a chance to hear about some of the work that's been happening over the last couple of years in an international effort around precision diagnostics. And then I'm fortunate to work with experts in diabetes technologies through my work with Rayhan, and with new insulin formulations through Danny Chou. And keeping us all seeing what's really important is Ananta, who has really led efforts nationally to think about health disparities in the context of diabetes. So we're hoping to weave together today updates on all of these important areas, and to think about it in this month of November, when we should all be thinking about diabetes and making sure that this is an equitable platform moving forward for all people, not just in the US, but internationally with diabetes. So I'm going to hand over to Ananta, who's really going to give us an overview of the foundational principles to understanding health equity through the lens of diabetes. So over to you, Ananta.
Thank you, Anna. And we're hoping to have this be a dynamic tag team, so you'll never quite know who's up next to speak. And but I think before we really dive into thinking about how genetics, insulin formulations, and technology can help or hurt the advancing of equity, I think there's a few foundational principles I always like to start with. And so the first is actually a rather old figure. It just turned 21 years this year, put out by the Institute of Medicine, so all the way back in 2002. But it's one that I often start with because I think it's an extremely relevant framework. And all that the Institute of Medicine posited when they did a sort of authoritative review on the state of current disparities all the way back in the early 2000s, and it still checks out now, is that the quality of healthcare, hopefully you can see my mouse, is definitely different between those individuals who are non-minority versus those individuals who are minoritized. Now, there's many ways you can define whether somebody's minoritized or not. Most commonly, and in this presentation, I'll be focusing on race, ethnicity, and socioeconomic status, not to conflate the two. Those are two separate, independent, and often compounding risk factors. And what they posited was that there is a difference. There's a clear difference. The data are unequivocal. And they said that they wanted to kind of denote, though, there's a disparity and there's a difference. And they said the disparity is really how healthcare systems and the environmental factors deliver healthcare. They call that the ecology of healthcare. And then, of course, sort of what we typically think of as disparities, which is around discrimination, stereotyping, and biases. But they said that something around clinical appropriateness or need and the patient preferences, that is not a disparity. So for example, I currently do not have any abdominal pain, but if a surgeon said, hey, I'm really interested in taking out your appendix for no reason at all, that might not be exactly appropriate. That's neither clinically appropriate, nor would it be what I would want. But a lot of the work that we're trying to do is really say, it's true that there are certain times when there is definitely not an indicator. But what we think as clinicians and as researchers and as sort of people in the medical community on what's appropriate and what families and individuals living with diabetes perceive, there's actually a fair amount of systemic inequities that come to those decisions. And I'll show you some data as we go along. And the second is we're going to talk a lot about disparities. And I'm a real keep it simple kind of gal. And to me, this is the simplest, most straightforward definition of what it means to have a disparity. And the disparity, I remember loving Venn diagrams when I was first introduced to it as a child, and I still love them now because that overlap of where efficacious treatments and inequitable access meet, that is where disparities exist. And so I think when we're thinking about understanding and operationalizing disparities, I keep my definition very simple because it helps me stay laser focused on where the work needs to be done. So where do disparities occur in diabetes? And this is a paper that's now around 7-8 years old, but it's around the presence of figure A is a map of the United States, but this pertains globally. I'm going to pick on the US a lot because it's where I live and it's where I work, and it's easier to throw stones at your own house. But again, it's relatively common trend. Panel A shows you the number of adult endocrinologists that are available, with the yellow dots being clinical centers, the dark blue being areas where you can typically access, although there is a lot of nuance there. And then the lighter areas, particularly in the Midwest, is where there's quite a few endocrinologists available. And panel B is specifically looking at pediatric endocrinologists. So really, and there's been a fair number of data and kind of analyses that look at the current workforce and the fact that there are truly not enough endocrinologists to go around to offer subspecialty level care. So first, there's a disparity in who gets to access subspecialty care. The second is one of the most fundamental things. We're celebrating the 101st year of insulin discovery, and insulin is still not reliably accessible to all individuals who need it and who need it to survive. The most recent statistics are something on the order of 1 to 4, or every 1 in 4 or to 1 in 5 individuals who live with insulin requiring diabetes ration their insulin. And this tends to increase if you're in that donut hole. So if you're somebody who's underinsured or uninsured, the likelihood that you're rationing insulin is significantly greater. And these same things play out by race, ethnicity, and a lot of the ways that you might see inequities. And the third place where there's a lot of disparities, and particularly very evident in the pediatric diabetes world, is around diabetes technology access. And these are currently all of the FDA approved devices that exist. Many of them are hybrid closed loop pumps, meaning that the sensors and the pumps all talk to each other. I can see the names that are in the audience, so I know you know this. And so you might also know, so this is now a paper that's 3 years old, where we worked with our colleagues in Germany, and panel A here is looking at CGM use, and panel B is looking at pump status, pump use. And then we compared those from the highest socioeconomic status on the left side of these bars to those of the lowest. And we have 2 time periods, light blue being the earlier time period and dark blue being the later time period. And what you can really appreciate in the United States is that there is a very strong socioeconomic relationship with who uses diabetes technology. And much of this is driven by how we deliver our healthcare. And what you can hopefully appreciate is, although there's been an increase, for example, let's take a look at CGM, in sort of technology utilization across the time period, you can hopefully appreciate that the highest increase has been in those kids who have, and this was a pediatric study, those kids from the highest socioeconomic status, whereas as you go across, it's a lower. So there's been a preferential uptake of technology. And coming back to what I said, there's inequitable access. So hopefully this shows you the inequitable access component. And again, I'll pick on diabetes technology, but then there's also the efficacious intervention. And one of the simplest ways that we look at efficacy in diabetes is hemoglobin A1C improvement as we're increasingly understanding as a crude marker, and it's also not whole person, right? It doesn't take into account well-being, sort of family stressors, and all the other things that mean living with a condition. And what this has shown is that you have an interval kind of worsening actually across all socioeconomic strata in the United States, but a preferential one, again, in those kids of the lowest socioeconomic status, where hemoglobin A1C has really increased. And then in a similar paper using type 1 diabetes exchange data, you can see here the various race ethnicities and those individuals who identify as non-Hispanic, Black, and Hispanic have a much higher A1C than their non-Hispanic white counterparts. I present both the socioeconomics and the race ethnicity data together because I want to be intentional about the fact that we're discussing race as a social construct and not a biological variable, because much of how the United States captures and records race ethnicity is that lived experience of what it means. So now I've hopefully proven to you that there is this inequitable distribution, and has very clear, in the most basic of sense, hemoglobin A1C impact. So now I'm going to take it over to Anna, who's going to help us tie in what it means to think about precision medicine, genetics, and why getting the diagnosis right is so important.
Thank you, Ananta. So yes, I thought it might be interesting to reflect on the last 3 years of my life, which along with about 200 other colleagues from the diabetes field has meant a lot of Zoom calls to work on something I don't think I'll ever do again, which is a systematic review. So this is a photograph of some of us who were at the European Association for the Study of Diabetes meeting in Hamburg last month, where Paul Franks delivered the results of the second international consensus report on gaps and opportunities for clinical translation of precision diabetes medicine. And this really was a Herculean effort. It looked at type 1 diabetes, type 2 diabetes, monogenic diabetes, and gestational diabetes. And we had 16 groups in total looking at precision diagnostics, precision prognostics, precision treatment for all of those different varieties of diabetes. And the idea was that we would work our way through the published data and identify what we really know about each of those areas of precision medicine, and identify some of the gaps and opportunities moving forward. And as I started at the beginning, we now have different treatments that are available. So it's even more important to be able to use those effectively in all people with diabetes. So my area of expertise is human genetics. And let me see if I can. Oh, I need you to change the slide, Ananta. I'm trying to move it and I can't. I was one of the co-leads on the effort to understand where we are, what is our understanding of precision diagnostics for monogenic forms of diabetes. Now, it's very important, particularly in children, to identify patients who have rare monogenic forms of diabetes as opposed to type 1 or type 2, because there are clear preferential treatments for individuals with monogenic diabetes, and also very important information for family members and for the prognosis of a patient based on their underlying genetic etiology. So I work with a wonderful group of individuals who are here shown on this paper from all over the world. We had Rinky Murphy from New Zealand. We had colleagues Paul Nolstad, Jan Mols, Pernilla Salvatoga, and Ingrid Alchrist from Bergen. We had Tony Pollen and Kristen Maloney from Bethesda. Some of you may know Tony leads the ClinGen program for monogenic diabetes gene and variant curation. Kevin Colcuff, Lisa DeFranco, Sarah Flanagan from the University of Exeter in the UK, Liana Billings from Chicago, Shivani Misra from Imperial College London, Catherine Owen from Oxford, and myself and Jen Ickley from Stanford. And as I said, we were meeting monthly for the last 3 years to really work out how we were going to tackle understanding what had been published by ourselves and colleagues and what we really know about how to deploy monogenic diabetes testing. And I think it really came down to some key guidelines that we could deliver around who should be tested for neonatal diabetes and monogenic diabetes, and how we should do it, and what the impacts would be for how those patients were treated. And that's summarized here in figure 2 of our paper, where we were able to really show that there's very compelling evidence that if you're diagnosed with diabetes, in the first 6 months of life, you should have a test for monogenic diabetes, including a gene panel with all neonatal diabetes genes. And the importance is that if you identify a mutation in the genes encoding the KATP channel, you can be transitioned from insulin to sulfonylureas. And this is an important thing to do because there is now a body of evidence that shows that that transfer not only improves your quality of life, but it also improves your glycemic control. We were also able to find strong evidence for screening women with gestational diabetes for mutations in the glucokinase gene. Again, really important clinical implications here around management of the pregnancy and whether or not you should use insulin treatment. But I think relevant for today's conversation is the lack of data that we were able to identify on comprehensive studies in non-European populations. So as we started to formulate the guidelines around the existing data, we could do that fairly effectively in European populations, but there were no studies in a large cohorts of non-European populations. And this is a real missed opportunity and is opening up for health disparities as we move forward with precision medicine, because we don't have the knowledge to understand what DNA changes mean in some of these genes involved in monogenic diabetes, which makes it very difficult to interpret them because we just don't have that background information on a diverse population. So if we could just flick to the next slide, Ananta. And here you can find in the section of our review, basically one of our recommendations here that an area of urgent need for funding and for research is to really understand what diabetes looks like in non-white populations for monogenic diabetes. It's very difficult to get that differential diagnosis right when many of the overlapping clinical features between type 2 and type 1 diabetes and monogenic diabetes are very blurred. So I think this will be an area of continued activity, and it's one that we'll be working on very hard for the next few years. And the next slide is about one of the ways that we're hoping to close this gap. And it's about trying to make sure that we can interpret the human genome at nucleotide resolution. So imagine you have a patient that you've referred for genetic testing, and you suspect that this individual has a mutation in a monogenic diabetes gene rather than having type 1 diabetes. But the genetic diagnosis comes back with a variant of unknown significance. And that's because it's never been seen before, and people don't know how to interpret it. And that can be because we haven't sequenced enough genomes from people from non-white populations. And one of the ways that we can accelerate how we understand those nucleotide changes is to put down detailed maps or atlases on the effect of those variants on the protein of interest. So I'm part of the Atlas of Variant Effects, which is an international collaboration to try and annotate the genome at nucleotide resolution in all clinically actionable genes. And I'm interested in doing this in the genes involved in all forms of diabetes. And the idea is that we make libraries of every single mutation in a protein of interest, and then we study it and the consequence of that mutation in an appropriate cellular context, so that we can make a variant effect map that can help us understand whether or not a novel change that's been found in a patient is responsible for their diabetes. So this is one way that we can sort of help to close that gap, because doing this across the whole protein is blind to whether or not that variant has been found in one particular population. So I'd be very happy if people are interested to talk about how this works. It's obviously a very ambitious project, but I think it's one that has the potential to really make a significant difference to clinical interpretation of diabetes genomes.
Can I just interrupt there for the Atlas of Variant Effects? You know, we can see the authors on the publication, but where are those data coming from internationally?
The Atlas of Variant Effects has thousands of members, and we are international. We have groups across the world. There are understandably strong contingents in Europe and in North America, but we also have membership in South America, in New Zealand, Australia. There's a lot of interest in Australia around working with Aboriginal genomes, and we have engagement across Africa as well. So one of the things here is about how we can also enable colleagues in other countries to set up the technologies and to be able to work independently rather than have helicopter science from those of us in other parts of the world. So this is a perspective that we wrote that is at the front of a whole edition of Genome Biology, which then provides maps on a whole series of genes, one of them related to diabetes, which is our map of glucokinase, which many of you will know is the rate limiting step for glucose metabolism in the beta cell and the underlying cause of one of the forms of monogenic diabetes.
Great. Yeah, thanks for that.
Okay.
All right, everyone. Well, Anna, that was a great overview. As a person living with diabetes who has 2 siblings with diabetes, I want to talk about how all of this affects the patient journey. And one of the big important factors is, you know, providers have significant biases. And one of the unfortunate things is this becomes particularly true for diagnosis. Oftentimes, we may jump to the conclusion that somebody has a particular form of diabetes just based on heuristics that we've picked up over time. And I think Anna's work and the desire to sort of try to catalog as much as we can, try to come up with methods and procedures for dealing with every population in an equitable manner, so important to getting the right diagnosis. And that's huge, right? That affects what happens to a person as they go forward. My work focuses more on automated insulin dosing and closed loop therapy for those who do end up having a diagnosis that requires them to be on insulin. Over my time as a clinician, I've realized more and more that glucose is just a reflection of the combination of life circumstances, socioeconomic status, and as such, we really have to work on tackling inequities if we're going to tackle diabetes care. So here is the current state of subcutaneous automated insulin dosing in the United States. You see, we have a multitude of commercial and open source options for treating diabetes with closed loop devices. We have 4 commercially approved automated insulin dosing systems, as well as 2 open source algorithms, which are implemented on both Android, iOS, and systems on chips. And it is important to realize that at every aspect of the dissemination and study of these technologies, there is disparity. So first of all, access to the components, right? So we have 3 components in an AID system: pumps, CGMs, and an algorithm that connects the 2. CGMs are largely unavailable in Africa, India, many parts of the world. And while it is possible to manufacture an insulin pump for around $100, actually doing that and bringing that to a jurisdiction is sometimes challenging, which reduces the possible access. So if you don't have a CGM, everything I'm talking about becomes irrelevant because you don't have the tools to drive an automated insulin dosing system.
And you can even, I would even offer that the CGM even improves, right? Just injected insulin for those who just have access to insulin and shots.
Absolutely, absolutely.
And just to sort of further that thought, you know, what percent of people in Africa and India have type 1? Those are huge markets for these types of you know, technology.
Yeah, so on average, we see about 1% overall of the population, and usually about 10 times that for type 2, of which some subset are going to also require insulin or have
And I'll just plug that some of the denominators of these countries are really large. So actually, there's quite a bit of work that a few colleagues in India have done where the N of India itself is so big that 1% of that population ends up being some of the largest absolute number of people living with diabetes. And then you're looking at trying to deliver care in an infrastructure that where healthcare delivery is a little, it's a Swiss cheese kind of situation. There's a lot of holes, there's a lot of variability, there's a lot of issues around access. But yeah, so I think there's a percentage, but there's also the actual absolute ends and the systems that they live in.
Right. Yeah. Real opportunities for improvement.
And it should be noted, right, that speaking of these denominators, so much of what we do is in the white overserved population, which is actually a minority population worldwide, right? There's the bulk of the world is not European. So this means that in terms of care delivery, first of all, access to the individual components is limited. When we are running the clinical trials for all of these systems, the population tends to be white, overprivileged individuals who get into the clinical trials. And this is for a variety of reasons. One of them being that at certain points, we've actually had restrictions on the studies. So you could not recruit the most underserved individuals due to A1C, due to the number of finger sticks an individual was willing to do in a day, due to other comorbidities such as psychological disease. So as a result of that, we recruit into our trials a very specific population, and we also are recruiting people who are largely coming in, following up with us, and that population usually does well no matter what tools you give them. These certainly make life easier, but when we label all of these equipments and technologies, they end up getting labeled for everybody, despite the fact that they were not studied in everybody. And on the one hand, that's great because we permit potential access to these devices, but on the other, we have to be very, very aware that we are not being equitable in the dissemination of this technology. And the other aspect, interestingly, in the US, is that when one of these products come to market, it almost always gets covered by private payers first. So the first people to benefit are folks with private health insurance. And then the public approval actually takes longer. So you'll frequently see, if you ever see data like real world data on any of these systems, you have to remember in the first couple of months of release, it's all coming from private payer insurance groups. And in a sense, this is making the rich richer. So
in just as an aside also, so the access though, right now is our CGMs, Dexcom, et cetera, and some of these AIDs available in the sort of publicly funded medical setting?
As a result of a lot of work that was done nationwide and some done at Stanford here, and Ananta can speak to this, we have borne out in the data that if you give someone a CGM, their diabetes control improves. And if you take it away from them, it worsens. Not surprising, but apparently we had to spell this out in the data in order to get payers to pay for it. The good news is that as more of that data becomes available, public payers are more willing to fund these devices. And so we are getting better and better coverage.
Better, but not everywhere. So I think we just looked at these data recently for a review that's coming out. And in 2019, around 15 states didn't cover any. And this is, again, US centric. So the disclaimer there, just because, but so 15 states
So
didn't
no
have
AID, well, none of
no
the
sensor. Actually, I didn't
no
even
sensor.
include AID because, like Rayhan was saying, if you don't have sensor access, AID is not even an, or, you know, sort of automated insulin delivery is just not even an option.
Yeah, sensor's
So
the entry point.
sounds exactly. And so around 15 states in 2019 didn't have any explicit, reasonable, reliable coverage. And now that's decreased to 7. And more states, again, because a lot of data that's being published, are trying to not just have it be for those individuals with type 1, but insulin requiring diabetes. So, but there is a lot of heterogeneity, both with state to state. In the United States, there is a good amount of data there, but also unfortunately so the legislature kind of tells you what the state has to do, but the regulatory aspects, which is how CGM coverage or insulin pump coverage is actually executed, that almost has actually county to county variability. So unfortunately, there's not a clear-cut answer we can give you, except that it is about clear as mud. And what that results in is a lot of interruptions, which, again, like Rayhan was saying, we've shown you don't just go back to where you were before you had your technology, you actually have an interval worsening in your A1C. So it really behooves us, and it's on us broadly, the medical system, to make sure that coverage, that you have access and that it's uninterrupted. But it's very heterogeneous, unfortunately.
Yeah, no, thank you for sort of like illustrating that.
And, you know, this is not just on the payers. When we charge far more than it costs to make a device or a drug, suddenly you are causing disparity. And then oftentimes we blame the payers, but there is a 3 order of magnitude markup on some diabetes drugs, for example, that is unconscionable, and of course is going to make people question whether they should or should not be covering things. Next slide, Ananta. This is just to show data that with one of the open source systems and a GLP-1, we can get people to a no meal announcement AID system. In other words, the person wears their sensor, wears their pump, they take a weekly injectable GLP-1, which reduces the spikes from a meal, reduces the quantity of meal that the person is eating. And here we see someone who went from bolusing twice a day to not at all. Their time in range went up despite not telling the system about what they eat. Time below range went down. And this is important because what it means is we have the technology to do AID without meal announcement. We still need to talk about exercise because that is a place where we still need to announce, but the technology is there, as Ananta was saying, but is it available to everyone? Hell no. And that's really the next steps in doing all of this is we can do a ton of stuff in the lab, but if it doesn't get into the hands of the people who need it the most, it's absolutely useless.
Yeah, it's a disconnect. And then for that previous slide, was that all diabetes or just type 1?
This was in particular a type 1 individual, but we could make the assumption that if someone was insulin requiring type 2, you might expect similar effects and certainly benefits to insulin resistance and insulin needs from GLP-1 therapies.
Right, thanks.
And so I just wanted to put a plug there. If anyone is interested in learning more about the open source algorithms, this is one where actually all the principles of biomedical ethics are fulfilled. Autonomy, because we know how open source AID algorithms work in and out because they're open source. Justice, because they're freely available on GitHub. Myself, Katharina Brown from Charity Hospital in Berlin, Sufyan Hussein at King's College London, and about 44 health care providers from 25 countries, 4 legal experts in diabetes technology, put together an open source AID consensus for those wanting to learn more about them.
Great effort. Okay, so now we're gonna
So I was on mute.
pass the baton here.
Okay, thank you,
Thank
Rayhan, for the great overview about the AID system and then basically the equity part for us to discuss. So I'm a chemist by training, and then we, as Monica mentioned in the beginning, we developed new formulations, and they could either be on the insulin side, or basically we add something inside the insulin formulation such that the whole thing can be more stable. And then just for some of you who don't really think about the insulin sequence or structure every day, basically we try to look at the insulin structure and then try to do some tailor-made efforts or activity in a way that I try to make something better. Next slide. So the big thing what we are trying to do is actually try to learn from the nature. So this is actually a review article co-written by myself and my former colleague in Utah, Helena Safavi. Basically, we do a bunch of different current and past efforts. People try to study what's inside the nature in a way that these could be some unexpected organisms, and then there are some amazing molecules for us to study. One of the very recent example and very well-known one would be the GLP-1 analog that Rayhan just talked about. And now that because of all the craze about Wegovy and the Ozempic, we all know that it's the first ever approved GLP-1 analog back in 2004 called Byetta, that was actually a molecule from Gila monster in a way that they share pretty high sequence homology with the human GLP-1 molecule. And then the original GLP-1 molecule has a terrible pharmacological property in a way that you will be degraded very fast when injected to human. But because of the similar sequence homology, the peptide called exenatide from Gila monster, it's active toward the human GLP-1 receptor, but also it can stay for a little bit longer inside the body. And then in the end, we know that a little bit longer is good enough to make it a drug. And then with all the chemistry efforts, they can originally, I think the exenatide was injected twice a day. A few years later, Novo Nordisk was able to do some more chemistry on top of it to have liraglutide or Victoza that becomes a once daily injection. And then a few years ago, they were able to do some even more sophisticated chemistry such that they can now extend the time to once weekly injection. And that's now making all the big differences, not just for glucose control, but also for weight control. I would say starting in 8 years ago now, we actually also have a pretty good example like this. Next slide, Ananda. Okay, so this is actually from basically fish hunting conch snails. They were originally identified in the bottom of the sea in the Philippines. So they are actually not that many of these kind of cone snails. Basically, I think in total there are probably 300 to 400 different kind of cone snails. They are actually all very well studied in a way that basically people probably study them as early as the 50s. The main difference, and then this kind of talk about how technology advance can lead to different discovery. So in the past, what people do will be they collect these cone snails, they chop it off, and then they will extract what's inside the snail, and then they will run through different purification techniques in a way, try to see what's out there. And because of the approach they use, they will normally identify what's the most abundant ones. So lots of times they will find, the main thing these snails use to kill fish, which will be neurotoxins or ion channel blockers in a way that they can intoxicate fish, because that's how they can do their prey hunting behavior. But just like what Anna mentioned early going, we can now do genome sequencing fairly cheaply. So instead of chopping things off, looking for what's inside the snail, one can basically sequence the genome, not on human, but now in snails, and in a way that people can now use bioinformatics techniques to guess what are the sequences out there. So in the panel C and panel D here, these are actually sequences we can now use bioinformatics to predict these different cone snail species in all these different kinds can potentially make. And actually, you can now see that they actually have very similar sequence homology with human insulin, and then potentially zebrafish in insulin as well. And this is basically the same analogy in the old days, how scientists see that the sequence of exenatide is actually very similar to the human GOP1 sequence. And this is basically exactly the same way we are looking for as well. The reason why we have to study them is actually because these are insulins that they have very short human insulin B chain. And this is actually very important here, because human insulin B chain, especially the B24 to B26 triple residues with aromatic groups here, they are very important for human insulin to engage with the receptor. Normally, if you basically cut this highlighted region off, you will actually end up getting an analog that's not active at all. So the potency will drop by 500 to 1,000 fold. So that means they don't even have any effect when you inject it in our body. And basically, what's special about all these venom insulin is that they actually don't need to have this region, but basically they develop all different things such that they can start to have different enhanced function without these regions. And this is exactly what we study in this paper, in a way that basically it's now a faster acting insulin. We demonstrate them in pigs in a way that basically, because it doesn't have this region. So normally insulin will form dimer and hexamer, but in this particular case, they are monomeric. So when you inject it in the body, they will be faster. And potentially this will be a very important behavior and property for the AID system, because right now, even the best fast-acting insulin like Linjef or Fiasp, it's actually pretty fast onset now, but the duration of the action will still make it relatively difficult for algorithm to make the best decision to basically make it to be even better, and then even better tightly control the glucose level. So what we are hoping is meaning insulin analogs like this can be helpful to further enhance the performance of the AID system. Next slide, Amanda. And this is the final slide I have, but I'm actually very interested and excited about this, because this is basically a project we started to do after I hear from Rayhan about some of the things we can potentially address in the AID system. So we all know that the delivery of insulin require cold chain delivery. So that means that normally insulin formulations, they are stored in 4 degrees Celsius. During the shipping, you will be shipped in a refrigerator type of truck. But what if we can make insulin to be much more stable and potentially make it even more concentrated, such that potentially we can deliver more, or for people who don't have access to cold chain delivery, it will be much easier for them to use. And basically, in this particular case, we decided to just with human insulin to go for it. And this is also part of the reason, because when you just use the human insulin, it's actually relatively cheap to access, because there's no patent problem. One can actually access Humulin from places like Walmart with very low prices. And what we are able to achieve here is we can actually make a U1000 stable insulin formulation. So this is 10 times more than what people normally would use in a U100 vial. And what we can do is basically in 37 degrees Celsius in constant shaking environment to basically mimic the aging situation, and in a way that basically we can make sure that in the upper side of the figure, so you can see that they don't get aggregated. Normally, when things got bad, they will form this kind of gel-like behavior. But when we add things in, you can see that they can just be a crystal clear solution here. And then we are able to basically test their activity in vivo, as you can see that compared to the 7-day age sample here, we can actually have a 6-month U1000 age formulations, and they are still as active as the fresh insulin right here. So this is basically another thing we are trying to address in places where it's actually very difficult to access cold chain delivery, and then these also tend to be places that don't have the best access of all new kinds of insulin formulations. And then we hope that this is something that in the near future that can be beneficial for basically, I'll say this is kind of more for third world countries where this is pretty difficult for them to access all the advanced kind of formulations. Okay, now back to Linda.
Thank you, Danny. And that's a perfect transition for one of the questions that also came up, which is what can we do? And it's around building systems. So there's actually in kind of the interest of time, because we all got actually quite excited and moved things a little bit faster, or talked a little bit longer than we'd planned, and I want to make sure we end on time. But I think I was going to take us through what the very, very first steps are to try to think about addressing some of these disparities. And I think we've made the pitch. I should expand the second figure to both say insulin and technology access. But sort of there are a lot of data. This is a figure for the Gatekeeper study that I led, and then that's since expanded to include from the Type 1 Diabetes Exchange, where it's very clear that the longer you've been in practice, the more implicit bias you have against public payers. And that makes sense, because clinicians have been raging against the system. Insulin and technology access is definitely making sure everybody has access. And part of that does mean payer coverage, but part of that means that people are getting recommended. And Rayhan alluded to this as well, is that inclusion into clinical trials is such an important component of it. And so what I'm going to do is I'm going to share with you one data slide of something that happened at Stanford, where we said every single individual youth because that's a pediatric center, who was newly diagnosed with type 1 diabetes within 30 days gets a sensor. And what I'm going to orient you here is, so this is the percentage of people who meet an A1C less than 7, which is the target. And in the gray bars, it's our comparator cohort. And then in the blue bars are those kids who are in this study. And what you can see, and this first one is stratified by insurance. This is just to prove that uninterrupted and universal access improves outcomes. So all of the kids who have public insurance in the light blue, or who identify as Hispanic in the light blue in the second panel, all did relatively better than their historical counterparts. And it's important to understand that just technology access alone isn't sufficient, because they don't quite close the gap, right, between their more privileged counterparts. So those individuals with private insurance and those individuals who are non-Hispanic still do sort of relatively better. But there's a interval improvement, particularly by the 12-month mark, where it almost looks like a staircase of improvement. And so this brings me to, I'm going to try to really wrangle everything that we've covered here to say, what is the path that a person living with diabetes, what is it that they travel in order to be able to receive the kind of healthcare that they would like, and frankly, that they deserve. And the very first thing that every individual has to overcome, and different individuals with different geographical locations, both globally and within the United States, different insurances, incomes, sexual orientation, gender, I mean, you name it, there's multiple ways an individual can be either hold privilege or be marginalized. And so the very first thing they have to do is they have to overcome their structural and social determinants of health. And that is in and of itself an entire topic. But if you think about Maslow's hierarchy of needs, if you don't address the most basic of needs first, we cannot even begin to climb this mountain that is the sort of the ideal healthcare. And then the next step is being able to, so that's even outside of medicine. That's things like being able to have transportation, some basic health literacy, just basic. And then we get to even further nuance, which is access to actual medical care. And that again varies globally and varies within the United States. And Anna talked about the correct diagnosis, the sort of literal face of how a person with diabetes, type 1, type 2, MODY, GDM, gestational diabetes, the literal face is changing. And it's really important that clinicians get the diagnosis correct, and that researchers are pushing people to think about what it really means. And then we talked about access to insulin. So that means that you're coming up with novel ways to store and continue to use insulin in areas where electricity is not reliable, or areas where the copay is so high that you can't actually afford all of the vials of insulin that you need. So if you look, I'm starting with very basic kind of base level things that each build on the other. And the next is, and I put here, not just access to diabetes technology, but utilization of diabetes technology. And it might be splitting hairs to use those two different words, but the reason is because access is a part of utilization, but then making sure, for example, if you have an older adult, that the text is big enough that they can actually read their CGM numbers, or that it's in a language that people can speak, or that it's in sort of the health literacy scope with it for various individuals. And a lot of that has to do with industry partners really making sure that they're making research and development component with as diverse of individuals as possible. And when you have all of those, you get an individual understanding and having ownership over their healthcare. And then they have the ability to advocate for personalized and precision medicine, which is where many people are sitting already. But I would posit that it's actually, our fourth or fifth step. And that really gets to flexible representative medical care provision, which is that much closer. Now, if you look, I haven't gotten us all the way to the flag at the top, which I'm going to call equitable healthcare, because there's probably a ton of steps still on the way. And so, but I wanted to lay a framework of depending on where the person lives, what their race, ethnicity is, income, insurance, et cetera, how they may or may not have choices over their own healthcare. And I think most people would agree, you should have the kind of healthcare that you would like. You should have the kind of diabetes management life that you would like. And so I'm gonna just kind of close with just an acknowledgement. I'll talk about the framework another time because it's 1 o'clock, and we want to thank all of our funders, of which there are a lot, and then also feel free to find us on social media. I think there's probably time for questions as well. But yeah,
there's time for questions, and I so appreciate, this is such a huge topic. As you've been unpacking it, we can see that there's room for, almost like each of you to do your own talk, because it's so important, and just kudos, hats off to all four of you and the important work you're doing in this space. And it's just amazing the progress that you have actually pushed forward in a short period of time as well. So just thank you all so very much. Was anyone like to unmute themselves or drop something in the chat?
I'd love to ask a quick question. I know that we are over time, so I'll hop off after this, but It was great to hear everyone. This is our team at Stanford. So I'm also working with this group and very excited to hear all the great research. I think one of the challenges as someone that's, kind of on the outside when I hear all of this important work that's being done, my focus is exercise. There's still a lot of importance. What would you recommend? Like, Ananta, I kind of think about you as guiding force of where do we go as a first step? I know it was kind of addressed in the comments as a question, but what's a first step that we can take to just, it's such a big hurdle. And that last slide you showed with just all these different avenues, do you have a recommendation for what can be the first thing we should think about?
That's actually the slide that I just breezed right through but yeah, I think most of us, if you're here in this conversation, chances are there's some involvement you have in the diabetes community. And there's a reason why you have it. It's because it's what moves you, it's your passion. And so, but there's little ways we can turn so that there is a sort of attempt at equity. So for example, if you're really interested in clinical trials, thank you, Danny, then making sure that you're recruiting equitably, that you budget for translators, that you budget for interpreters, and that you push your funders and you push your industry partners, right? So, and then if you're in the basic sciences, making sure that your genetic risk scores, making sure that that the data used to generate are as diverse as possible and are as representative as possible. And when they're not just put it as a limitation and say, oh, this limits generalizability and move on, but really push. And then the third thing is most of us are doing statistics, and there is a principle to, and there's quite a few examples. And again, we're out of time, but where you have to commit to dig deeper to the data. If the data say something, and that that's setting things up for inequities. Oh, this minoritized group does worse. Well, I would encourage you to dig deeper into that data to really understand what that driver is. So, because it's usually not the community, it's usually the way we've designed the instrument, or, and the fact that they haven't contributed to how the interface of, for example, a CGM tracing looks, or what languages are available in a pump, or how big. So, I mean, it's just really understanding that we've been driving this entire enterprise without input from individuals who now we're sort of penalizing, that it isn't speaking to them.
Yeah,
I think it's also important that people come to you, or people like you that are experts in this field, to even just have these discussions, because it may feel like I am doing something, to advance, where we're going with this and including maybe minoritized populations, but I'm not thinking about the underlying things that we could be doing differently, or, just discussing it with the people that spend their whole career focusing on this. So I think it's really important to get in touch with people like yourself.
That's a great point. I would also say, it just opens the door for more cross-pollination between sort of the hard sciences, the social sciences, the clinical sciences, and all those who really interface with diabetes and, overall, but specifically type 1 diabetes. I would also ask, just really quickly, I know we're running out of time and I feel like we should have another one of these. So we'll discuss. But, what is, it also seems like a big thing we didn't talk about is this is often a pediatric disease. And so there's the family, right? The family, and they are acting as, caregivers and clinicians in some ways, with this, you can die with insulin. It's a very deadly drug if not used properly. So I think that, it's another huge component of how to reach the family members who are the primary caretakers for these pediatric patients. And I'd love to try to do another one like that in the future. So we'll talk again. But if you have sort of one little high level note on that.
And then I don't need to monopolize this, but so a lot of my early career work is on looking at sort of the family dynamics for individuals who hold one or more minoritized identity, and what is their relationship to diabetes care. And actually that's part of why I started with the structural and social determinants of health as the underlying. So their prior experiences, their generational experiences, and what's been passed down strongly influence whether they engage in research or not, or whether they engage in certain clinical care systems or not. And it's really important that we understand that in the clinical world, in the research world, so that we're really understanding where they're coming from, because there's very good reason for it.
Yeah.
And there's definitely, and then things like structural race, like implicit biases and structural racism, they cite that as reasons why they're very hesitant. And there's a good body of literature that also supports all of this. So
well, maybe
yeah,
we can
you're absolutely
unpack
right.
that in another session, and maybe draw in the T1D exchange, cohort or group, because they also, I know, are trying to understand that better.
Yeah,
In the interest of time, I just want to say thank you all so very much. This was really fantastic and so illustrative of all the work that you're doing at Stanford and how it's such a sort of really collaborative effort globally, extending to the globe, but also really thinking about the layers of type 1 diabetes. So important, really so important. Thank you all so very much. And people can reach out, I guess, directly to you as well. We have your, this will be posted on our website, and people can listen to it at their leisure, at the bench or on their drive or wherever. So thank you again. I hope you have a great rest of your day, everyone.
Thank you for the invite. Really good to be here. Thanks, Monica. Take care, everyone.
Thank you.
Bye, everyone.
Bye-bye.